So what experimental approaches do you use in your lab to study these huge RNP complexes?
We always start with biochemistry. I think most of the people in my lab, the students and the postdocs, I think they probably spend 80% of their time on the biochemistry. I think you have to understand the biochemistry to be able to get these machines and, to understand how the proteins talk to each other, how they interact with the RNA. And then from there, with this information, then we go structurally. From there, we go to crystallography. We collaborate with NMR people. We don't do it ourselves. EM, I think, is changing. And we very much look forward to going to this much more. Now we have a facility in the institute with the EM microscope and the staff scientists, and many groups are getting into it right now. And so, as this coincides also with the complexes getting bigger and bigger. So, it's going to be interesting in the future to look at this.
Yes, so this is true. Some people have used this term lately, and it's probably not an exaggeration, because for a long time cryo-EM was still a very low resolution technique, so people were used to solve structures at 20 angstrom in resolution, and people called this also blobology because you could identify large proteins and their domains maybe. But with the recent advances that come basically from hardware improvements in the microscopes, but also more importantly even from direct electron detectors that make very efficient use of the signal basically, together with software, there have been now a burst in a number of really many high resolution structures, meaning at a resolution below close to 4 angstrom and below. And this is a resolution that enables you to build amino acids into the cryo-EM density, and this is then in the end very similar to X-ray crystallography, where this is commonly done. And so now basically cryo-EM is on level with X-ray crystallography, and then it just has the potential that you don't need to crystallize samples, and therefore many samples so far that were interesting, don't readily crystallize, and therefore it's a great technique to study large assemblies.
Interested in X chromosome inactivation and long non-coding RNAs and the role they play in that process. So we're using state-of-the-art genome engineering first to understand what are the consequences of loss or gain of function of those long non-coding RNAs. We do a lot of imaging also to try to see how they are expressed, how they localize in the nucleus, and we do some more biochemical, let's say, assays to try to understand where they bind and how they are regulated.
More recently, we focused on mammalian systems, trying to understand what's the role of non-coding RNA in the complex regulatory circuitry which control muscle differentiation. So muscle differentiation is a very nice model to study differentiation, since you can recapitulate in vitro all the different stages of muscle differentiation, from stem cells to mature muscle fibers.
I really got in love with the possibility of combining molecular biology with imaging approaches, especially live cell imaging. And that's what I have been trying to develop over the years. And I'm very excited to be today at the stage that we could finally been able to visualize in real time in live cells the processing of a nascent RNA. So by being able to fluorescently label nascent RNAs, we can track the lifetime of an intron. The results that we obtained indicate that introns are very short-lived. So we see the disappearance of an intron from a pre-messenger RNA occurring within seconds, which was a surprise because this goes against many of the previously published data indicating that splicing would take a couple of minutes.
What experimental techniques are you using to study myotonic dystrophies, and I mean, what are the challenges working with these RNA repeats?
Okay, that's a good question, because since there's not a protein translated from this repeat, we are at the level of RNA, so it's all RNA-based techniques. Fish, of course, RNA fish, the detection of the fish in patients' tissue, which is not that easy. And afterward, it's very common techniques, more physiological techniques to engineer, to develop models, because we cannot work on these poor kids. We can have cells or tissue, sadly, when the people are died. But so we develop models, fly model, Drosophila models, mouse model, rat models. But the importance is to develop models. So we express this RNA in these organisms, and we studied this, for example, this mouse model to see if they reproduce the symptoms. And if they reproduce the symptoms, we have now a model to test drugs or treatments.
Made a transition to doing high throughput type work back in around 2000 or so. Our goal was at that time was to go beyond looking at alternative splicing on a gene by gene basis and try to understand more globally how it's regulated, and that motivated us to develop custom microarrays along with our collaborators to figure out systems that we could use to profile splicing on a large scale. And from that technology, we carried on improving on it, and then RNA sequencing came along, and from using RNA sequencing analysis, we've really sort of expanded our scope of monitoring regulation across different cell types and conditions, and that's our method of choice currently.
This raised the question, is this an artifact? Because we are looking at reporter mini-genes. And that's what took us to try complementary approaches to address the timing of splicing in a global genome-wide manner that would be independent on any type of manipulation. And that's why we started to look at NetSeq data. NetSeq stands for Nascent Elongation Transcriptional Sequencing. It's a genome-wide approach that captures the nascent RNA that is still associated with the polymerase. And what we found that we think is extremely exciting is that we could detect spliced RNAs already attached to the polymerase, and we could map the position of the polymerase when splicing is occurring.
What do you think was the biggest leap forward, though, in terms of techniques in the last few years?
I think that RNA-seq has really helped us quite a lot in identifying very low abundant species, and also to identify these new species like circular RNAs. So that really helped quite a lot. And then on this new technology of cross-linking RNA and proteins and purifying these complexes.
What are the advantages of iClip compared to other clip techniques?
Right, so the iClip is the method we developed in Cambridge together with Julian, who was here at the conference. And the method really allows to amplify the full range of cDNAs that are produced during the reverse transcription when we convert the RNAs bound to a protein into the DNA complement, so we can then sequence them. And the real advantage is mainly that it is allowing you a greater coverage of the binding sites, and also because you identify the cDNAs at truncated position where the proteins cross-link to the RNA, you can use those cDNAs to pinpoint the exact position of cross-linking. So this high resolution can also be very useful to understand exactly how different proteins assemble relative to each other. This is again very difficult to model in vitro because proteins often contain sequences that are difficult to synthesize. They can behave very badly in solution, they can precipitate and so forth. So again, having an in vivo environment, just cross-link these protein-R interactions in vivo and then purify them to genome-wide analysis allows us to actually gain first insights into how these proteins assemble, make a hypothesis, we can then follow up with other methods.
So with this crosstalk and interconnection between networks, how do you go about, or what experimental techniques do you use to to dissect this and to...
So the idea
understand
is that to use a systematic approach, a high throughput approach, by which we systematically knock down every component of the splicing machinery or every regulator that is known. And then we systematically look at the effects of each of these knockdowns, which basically reduce the levels and therefore the activity of these proteins, no? On a series of alternative splicing events that are functionally related. For example, they control cell proliferation or control programmed cell death. No? And then what you do is you try to capture from these profiles of alterations in splicing that you achieve when you knock down one factor. You try to establish a profile of perturbations and use that as a sort of a phenotype. And then you compare how the profile of splicing perturbations by one factor, how similar or dissimilar it is to the knockdown, the effects of the knockdown of another factor. And so then factors that are very functionally similar, they will produce upon their knockdown a very similar profile of alterations in splicing. And then this means that these two factors are going to be functionally connected in this network.